alluded to it earlier that I'm not a huge fan of a one size fits all. So as I said, the theme of the trainee talks, and we kind of did handpick them through the PIs, is let's get away from one metric collected with one piece of equipment in one lab. And so we've got 5 different talks that are kind of going to look at it from different aspects. But that's kind of the theme of the talks from this morning. So our first talk is from Dr. Jessica Cooper, who is currently a postdoc and fellow at UBC, working with Dr. Dave Wilson. Prior to this, she was a PhD student in Dr. Jan Wronski's lab at University of Calgary. And her presentation today is going to highlight a portion of her PhD work along the lines of imaging and how that can fit into, as you can see in the slide here, an integrated systems approach to investigate pathomechanics. So Jessica, over to you.
Sorry, I realized I was still muted. So as was discussed in the debate, a variety of potential factors, including aging, obesity, and injury, are associated with changes in cartilage homeostasis that interfere with the tissue's ability to adapt its structure and biology to its mechanical loading environment. Consequently, a progressive degradation process is initiated where abnormal repeated loading and altered cartilage structure and biology interact. In the Wronski lab, we are interested in linking the changing mechanics to the structure and biology of cartilage. We can start by looking at the healthy state of cartilage and one of the risk factors, aging, to better understand this link. This leads to our research questions. We have focused on what is the relationship between cartilage contact mechanics and cartilage composition during dynamic movement, and how does aging influence that relationship? We have focused on a variety of areas, including looking at cartilage contact mechanics, such as proximity mapping of the cartilage, contact paths, and sliding distance. We can also investigate cartilage composition using T2 relaxometry, which I will describe further. Today, I will be focusing on showing how we have linked the last 2 areas in an integrated approach to answer the research questions. T2 relaxometry is a magnetic resonance-based measure. An increase in T2 reflects an increase in free water content in cartilage, and this has been linked to decreased cartilage stiffness and increased permeability. In aging participants, T2 has been found to be increased in the superficial layer of cartilage. And the question we ask is, does the T2 remain constant over the regions in contact during gait? We'd like to understand whether higher and lower T2s correspond to the movement. So we recruited 5 20 to 30-year-old and 5 50 to 60-year-old male participants. Both groups were asymptomatic for OA. We integrate a high-resolution MRI scan shown on the left using a 3T GE scanner, which allows us to segment models for bone and cartilage shown on the right. To get in vivo bone kinematics, we're using high-speed biplanar video radiography, or HSVV. This allows us to view the bones moving dynamically from 2 views. We can measure position with sub-millimeter accuracy. Our data were captured for 0 to 25% and 75 to 100% of the gait cycles, so a portion of stance and a portion of swing. One of the reasons for this limitation is occlusion of the limbs when they cross over and the field of view as well. Kinematics were determined with a 2D, 3D manual registration procedure where we align the bones with the images from the HSBV with the bone models from the MRI. And then the kinematics are applied to the cartilage models to obtain cartilage contact maps. Proximity method was used to determine regions of contact, and this contact was determined for each frame of data. A weighted centroid, as shown in yellow, was used for contact location and path, and those are shown on the right for stance and swing. With regards to T2 imaging, T2 weighted MR images with 16 echoes were manually segmented for femoral and tibial cartilage. T2 values were calculated on a pixel by pixel basis using a least squares fit. Having mapped the T2 over the surface of the joint, we could then register the T2 surface shown in pink to the contact surface shown in blue. T2 values from the T2 data were assigned to each mesh face of the contact model for both contact regions as shown in yellow and non-contact regions as shown in blue. We then identified the contact faces based on the proximity values less than 1 millimeter and found the average T2 over contact for a given frame. This plot shows the T2 average over the contact region on the y-axis against the gait cycle normalized to 100% on the x-axis for the younger group. T2 values remained effectively constant over stance on the left-hand side of the plot. In swing phase on the right, you can see that T2 values moved from high to low with a minimum at 90% of gait as the joint prepared for weight acceptance. T2 values over the region of cartilage contact and the gait cycle showed a relationship, and there was a consistent pattern of T2 across participants over the gait cycle. Looking at the older participants for the same plot, we see a similar relationship. Further, no differences between younger and older groups were seen at first force peak. We also looked at the medial tibial normalized medial lateral position of the weighted centroid. So looking at the box plot, we have the normalized position on the y-axis and the box plots for swing and stance shown on the x-axis. We saw relatively consistent location and low variability amongst the younger participants. In the same plot for the older group, the median medial lateral position was more variable, with larger variability within subjects compared to the younger group. When we looked at potential patterns between T2 variability and medial lateral contact variability, we did not see a trend. Here you can see that in the younger group, variability was primarily low in both categories. Looking at the older group within the medial lateral variability, there were higher magnitudes compared to the younger group, but still no pattern overall. So low T2 implies healthier cartilage supporting high levels of loading, such as during stance phase when greater than 200% body weight can be experienced. A relationship was observed between T2 in the contact region and the gait cycle. In particular, T2 was lower during stance phase. We can speculate that this may be advantageous and pose the question, does this mean that cartilage is adaptive to the local loading environment? No differences were found between younger and older participants with T2 at first force peak, and no clear associations were made between T2 and medial lateral contact variability. During this process, one challenge was joint normalization. Joint normalization approaches to enable comparison of results across studies and research groups. So one approach that we used for normalizing the joint here was maximum bony joint width for the weighted centroid, but there are definitely opportunities for standardization of methods across labs. We have been trying to move away from single outcome measures by integrating multiple systems, biological with mechanical, to attempt to associate multiple outcome measures. MRI and HSBV were integrated to successfully investigate contact mechanics with cartilage health as a function of the gait cycle for the first time. And I'd like to thank the following people and funding sources for their support. Thank you.
Great, thanks very much, Jessica. And I think that was a really great lead into this session. And as I mentioned earlier, again, the theme is let's do more. Let's start looking at ways that we can integrate different approaches, and I think you showed that very nicely. The other comment I'll make too is that you're using new metrics of cartilage, in this case composition, that I think are going to be very useful moving forward. So that's great and a great start. Looking forward to seeing more of this work coming out of both your current position as well as Dr. Ronsky's lab still at Calgary. So got some time for a question or two from the group. I don't know whether people, it's on your side, but I don't have a chat function or chat box on my side. So I'm wondering whether people just want to raise their hand if they want to ask a question. Okay, I'm not seeing one, so I'll just ask one. Again, you chose T2. Can are there other ones that you had thought about originally? T1 row, for example, or T2 star. What made you settle on T2?
So yes, we did consider T1 row. We did have some practical limitations with the availability of T1 row in our GE scanner.
Yeah.
But T2 has also shown promise as a measure on its own for cartilage composition. So we felt that it was a strong approach as well.
Great, thanks. And again, just another comment. I loved how you could kind of look at the changes at different regions and also through the gait cycle. So really great work, and as I said, a real good addition to the literature.
Thank you.
So moving on, our next speaker is Dr. Carrie Costello, who is currently a postdoc fellow at the Department of Physical Therapy and Athletic Training at Boston University, as well as the section of rheumatology at Boston University School of Medicine. She'll be presenting some of her work from her current position in Boston, as well as her prior work during her PhD in biomedical engineering at Dalhousie. And for those of you who don't know Carrie, she's also a big proponent of promoting biomechanics and osteoarthritis research from a social media perspective, podcasts and whatnot. So if you don't know Carrie by now, you should reach out to her and see how she can help promote your research moving forward. So Carrie, I'll turn it over to you.
Great, thanks, Michael, and hello, everyone. I want to start off, I know many of us are already aware of this, but osteoarthritis is kind of a unique disease in that there is a large part of it that appears to be mechanically driven, as we've heard multiple times before in this morning's debate. So if we can better understand how this joint loading is related to the disease progression, we may be able to intervene and alter that disease course, which I think is what we all want to do. But in order to do that, we need to be able to actually capture joint loading exposure. So ideally, I think we'd want to be able to measure across the whole joint so that we could capture variations in loading across tissues, as well as be able to measure continuously so that we could look at variations in loading over time or with specific activities. Now, I know you just heard a little bit about how we might be able to do that moving forward, but unfortunately, I think right now this is only possible mostly in specialized circumstances, such as an instrumented knee replacement. And that's not quite as helpful for trying to understand the loading in the native knee. So what can we measure? Right now, we can look at how someone moves in our motion capture laboratory, which gives us an idea of the joint loading magnitude or duration during a single typical step, for example. And then we can also try to look at what people are doing in real life by looking at physical activity patterns using wearable technologies, which gives us an idea of the frequency of joint loading and also how many of those steps they take in their normal life are actually those typical steps. And this actually gives us quite a bit of data. So for example, if we want to look at moments in EMG from 2 knees in our gait lab, and then we send the people home with an accelerometer for a week, this is going to give us almost 1 million data points just for this single person. And this may be somewhat basic selection of data that we'd be capturing in this example. So as you heard earlier today, what's traditionally been done is that this is often distilled down to a few key variables. So from our 3D time varying joint moments, we might distill this down to a peak value from a single dimension. Or similarly, those 3D accelerations from 7 days might get distilled down to a single summary metric like average daily step count. And while these have led to some interesting insights, there's clearly a lot more information here that's not currently being utilized. We may be able to gain new insights about OA or new avenues for conservative management targets by investigating whether there's additional value in these data. So again, as you've heard a bit about already today, one way that that's been done specifically for gait data is through using principal component analysis to look at patterns in the data rather than merely discrete metrics like a peak value. So in some work at Dalhousie that was led by Dr. Gillian Hatfield, as you again heard about a little bit already today, this approach was used to identify pattern differences in both joint moments and EMG at baseline between those who did and did not go on to TKA 5 to 8 years later. Specifically, a lack of mid-stance unloading in the knee adduction moment, less dynamic knee flexion moments, and prolonged mid-stance activation of the quadriceps and hamstrings. So there does appear to be valuable information in these gait waveforms beyond what might be captured with these traditional key metrics that we've heard about often. What about joint loading frequency? So during my PhD at Dalhousie, we looked at this in a small sample of individuals for whom we had TK outcome data at a shorter follow-up of about 3.5 years. And we actually did not see any differences in baseline step count or intensity-based physical activity metrics between those who did and did not later progress to TKA. However, in a cross-sectional analysis in a larger sample, we found some interesting relationships when we looked at gait differences across quartiles of step count. We found that the lowest quartile step count, the dark dotted lines in these plots here, typically had gait patterns that were different than one or all of the other quartiles. And moreover, their patterns looked more like those that were shown to be at risk for progression with less dynamic joint moments and prolonged muscle activation in mid-stance. So what does this mean? While it appears that those longitudinal results might suggest that gait is more relevant to progression than some of this physical activity data, we also may not be capturing these complex relationships between gait and physical activity that we see in the cross-sectional analysis by looking at these different variables individually, or potentially even if we looked at them in linear combinations. But overall, there does seem to be more signal in these data than just those few key metrics, and this could be valuable to understanding progression. We see relationships between gait and progression, possibly also something going on between physical activity and progression, but maybe through a complicated relationship with gait patterns. I also didn't talk about it here today, but we also see differences in these relationships depending on how we define progression. And then as Janie nicely laid out earlier, another concern is that the OA population is very heterogeneous. And the traditionally small samples for gait data have kind of limited our ability to try to account for both that direct effect of these confounders on knee osteoarthritis progression, as well as the effect they have through changing gait patterns or physical activity. And then of course, there's always the question of how to deal with walking speed. So it seems that we may need fairly large data sets to explore all of this, but even if we did have those, it's still quite challenging to model all of these relationships in a traditional statistical analysis. So how can we put this all together and try to make sense of it? So during my postdoc at Boston University, we've been experimenting with using some machine learning approaches to look at some of these questions about how these inputs are related to progression, because these approaches don't require you to pre-specify all of these underlying relationships. So I'm just going to share one recent study we've been working on, which utilizes data from the multi-center osteoarthritis study. And this is a large cohort study of people with or at risk for knee OA. Because of the clinical setting, they don't have full motion capture data available. However, ground reaction force data and accelerometer data from a week of wear were available from this fairly large sample size relative to traditional gait analysis. So we wanted to use machine learning to see how well we could predict medial cartilage worsening damage using a combination of features extracted from the ground reaction force data, accelerometer data, and also demographics and clinical characteristics. We had a sample of over 1,000 legs, of which 14% experienced cartilage worsening in our 2-year follow-up. And using an ensemble machine learning approach, we had a median area under the curve of 0.78 on our test sets from 100 random data splits into 70% training and 30% testing. So we thought that this was pretty good and shows promise for machine learning prediction using gait and physical activity features. However, also of interest is that using a variable importance measure statistic, we identified a number of these ground reaction force and accelerometer features that were frequent top contributors to this prediction of progression. It's still not totally clear from this how these different variables relate to progression or whether there are any interactions among these different variables, but this does show promise. So to wrap up here, I just want to present what I think are a few remaining challenges and future directions to try to stimulate some discussion in the spirit of this symposium today. So first, in our most analyses, we are planning to move towards using some deep learning algorithms on the entire ground reaction force waveform and eventually the accelerometer waveforms as well, so that we can try to utilize all of the available data and try to capture some of these time varying patterns of loading that might not be as possible to get at with discrete metrics, even when we use a wide selection of them. Second, this came up earlier today, but I think it's going to be very important to try to capture joint loading in more realistic conditions, since people are often not engaging in a range of activity, or they often are engaging in a range of activities during their day, and not just that typical step or activity that we're capturing in our motion capture laboratory. So there's been a lot of nice work coming out in this area recently, and this could give us a better picture of the actual loads the joint is experiencing in daily life to improve our ecological validity. And then last, as I just mentioned, identifying intervention targets from these more complex machine learning models is still a challenge. We may be able to use some newer machine learning approaches to, for example, identify which parts of certain waveforms are important to progression. So maybe we will get back to some key metrics, but they may be different, or there may be a wide variety of metrics that we decide are important. And then similarly, I think we still need a way to figure out some of these underlying relationships, particularly between gait and physical activity, because we may actually want to target a combination of these variables to try to optimize OA outcomes. And this could even vary per participant or patient, as we saw earlier today. So I'll just leave it there for now. I'm excited to hear what you guys have to say. Just want to acknowledge our study participants, my colleagues, and also our funding sources.
Great, thanks very much, Carrie. And I see Dan Ramsey's got his hand up, so go ahead.
Hi, Carrie. Thank you very much for a really interesting talk. I'm really curious about your wearable devices, in particular the accelerometers. The reason why I ask is we wanted to seek funding on looking at knee braces and putting accelerometers on the brace, one, to track wear, and two, looking at their physical activity count during the day. So my question to you is that what device did you use, and where was the wearable position? Was it on the wrist? Was it a wristwatch, or did you actually track the movement of the knee?
Yeah, so that's a great question. And I think, again, something that our community needs to think about more, depending what we want to get out of those accelerometers. For the two studies I presented today, there were two different accelerometers used, the one at Dalhousie and the one at BU, but they were both worn at the waist. So they weren't at the knee. I think Dylan Kopsar just put out a nice review on using wearables for gait analysis. So I direct you there if you're looking specifically at trying to get at gait variables, but there are a number of different ways you can use those devices that I think are really exciting.
One other question, Carrie, is that I know that the battery life of some of these devices are pretty small. And I was wondering, I mean, I think it's really contingent on your sampling rate. So what was the longevity of these devices before they had to get returned and recharged and the data downloaded and whatnot? Because it's my understanding that there are reliability issues as well. So if you're sort of swapping devices to different individuals, then sort of the data gets washed out. So I was just wondering how you were able to control for that.
Yeah, that's a good question too. Again, I'm not an expert on these wearable devices per se. Both of these ones that were used for these studies had battery lives of a couple weeks, and they were given out for a week at a time, and then they would get charged and reset before given out to the next participant. But that is an important thing to consider if you're planning to use these types of devices in your research as well.
Thank you.
Great. Thanks very much, Carrie. It was a great talk. And again, theme of the day, let's see what more we can do and not just focus on one thing or one construct. That's great. Next talk is from Jesse Charlton, who's a PhD candidate at UBC and working in my lab. He's initially interested in biomechanical consequences of gait modification for knee OA. His current work is kind of, again, following on from the last talk. looking at using wearables to measure walking and free living conditions, and how we might be able to combine data from multiple sites. He's expecting to finish in the next 12 months, and he's kind of looking at what his next steps are, no pun intended, and how we can, again, be more impactful with our studies by being more collaborative. So Jesse, I'll turn it over to you.
Awesome. Thanks, Michael. Hope everyone can hear me and see my screen. I'm going to take a slightly different approach this morning. I think we've seen some really cool presentations so far on particular outcomes or things that we can measure. I'm going to pivot a little bit and look more at kind of the broader methodology that we use. The theme that I wanted to talk about is stepping beyond that traditional biomechanics framework, and I'm certainly a fan of memes and of Monty Python, and so we might consider the proverbial angry white rabbit as the traditional framework, and we're going to away from that. So generally speaking, when we conduct biomechanical studies, we do them in single labs. We tend to have 1 or 2 pieces of equipment in that lab that we use. And there's good reason for it. It's certainly quicker when you have everything in one space. You know how to use it. You have research assistants or graduate students to run it for you. And so you can conduct studies quite quickly, or at least quicker in relativity. You also have really convenient oversight. Everything is in one spot. You can see how the study is progressing, make changes, pilot stuff quickly, and that's really important. One of the problems, though, with these single lab research studies is that we have quite small samples a lot of the time. And that's quite pervasive across the biomechanical space, is that our sample sizes are generally low. This often leads to questions of diversity issues, where we have few individuals from different populations within the general category that we're trying to select people from, in our case, osteoarthritis. The other issue is that because we only have a few pieces of equipment or a few skill sets in our individual lab, there's only so many outcomes that we can actually collect. Now, what I would suggest is that if we move towards this more collaborative approach, we can solve one of the big problems in biomechanics studies, and that's sample size. And Monica brought that up earlier, and is a really, really important point. If we can collaborate and bring multiple labs together into a given study, we can increase that sample size quite quickly. But more importantly, we can get better representation of the people we're trying to study. For example, at our lab, we often have individuals who are more affluent, mild, moderate knee osteoarthritis who come to our studies, partly because of our geographical location, our various different recruitment mechanisms. But if we collaborate, we might be able to get younger individuals who have early OA. We might be able to get individuals who are later into the disease stage or on a surgical wait list, for example. Another piece with these collaborative studies is we can combine resources. We can use the money and the personnel and the expertise spread across multiple labs and years of experience to come together and improve the study overall, and potentially reduce the financial burden on a given lab or institution. Of course,